Okay, so this is going to be the video that talks about how we use the pre-processing scripts that we currently use in our lab, how to run them, how to organize your data for them, and how to understand what they're doing and what's coming out of them. Now this is all current as of January 2019, so if you're from the future watching this and it's significantly past this date and you're trying to learn this script stuff, so maybe ask the more advanced students in the lab who are using fNIRS if anything major has changed to this. And if you are that advanced student in the future, maybe make an updated video to this to replace this one. But for now, I'll show you how we're doing things at this current time. So we use right now a package of scripts written in both Matlab and Python to pre-process the data that's coming out of our fNIRS machines into oxy and deoxy concentrations that we can then do analyses on. Right now that data lives at my GitHub, smburns47/preprocessingfNIRS. This is the URL up here. And so to get it, first you just have to go to this page called the repository page and either download this as a zip file where you can then unzip the files and get all the scripts and everything, or if you are handy with Git on the command line, you can use this as the URL you put into your Git command. When you do download this kind of collection of scripts, the first level will be two directories that contain the Matlab and the Python version of this code. This video is just going to be talking about the Matlab code. I'll make another video that talks about using the Python version, because you only need to use one of these. The Python is just pretty much a clone of the Matlab version that was written first. In the Matlab version, there are a few scripts that this pre-processing pipeline depends on. The first one that you're going to be running and that will be using all the other scripts at various points is this one called preprocessingfNIRS. Has the same name as the script itself. Or sorry, the same name as the entire download that you just made. This is kind of a wrapper function that handles all your data and your different files. It's possible to take a full directory of your data scripts, run this once, and then it pre-processes all of that data, so you don't have to do it subject by subject. You can just do it as a batch process. And then within this one, it kind of handles variables for the data and calls these other functions at different points. And as we go through what this does, I will show you these and talk about what each of them do. Now at the top of this preprocessing fNIRS code, I put lots of instructions and comments and debugging tips. So you'll want to read through that first to get an idea of, say, what inputs you need to be putting in, how the outputs come out, etc. Now, you won't be able to just run this straight off the bat from downloading this. It's built off of two external sets of code that I did not write. The first one is called inpaint-nans. That I did package into this preprocessing fNIRS download in the Matlab version because it's a tiny little bit of code, so that was pretty easy to add in. But it's written by someone else, so I just didn't want to reproduce that without, giving them credit. But then the other batch of scripts that it will need, which are much bigger to wrap up into this, so which I didn't, is called Homer2. Now this is a set of pre-processing capabilities that the multifaceted team over at the Boston Hospital wrote. Major contributors are David Boas, Ted Huppert, Maria Franceschini, and they kind of one of the first to develop a really big functional package of code that can handle fNIRS data and turn it, the raw values that you get out of a machine, into something usable that then we can analyze. So they are still producing these and updating it every once in a while, so that's why I wrote this as a dependency instead of a package that's wrapped up into this. So you'll want to download Homer2 script. that just lives at the Nitric web page. So let's look for that. Here. Make sure you get Homer 2 and not Homer. That was written in the early 1990s and is not what you want to use. Homer 2 will have a much, much bigger capability and has the code that we are using. So make sure you download this and unpackage it and put it at least on your file path in Matlab, or if not in the same folder that your preprocessing directory is in. Okay, so using preprocessing fNIRS. It takes two arguments, a data prefix and a Boolean that tells you if you ran dyads or not. So the data prefix means when we have all our data, it's going to be structured in either folders that are for each subject and then all of the scan files in that folder, or folders for each subject and in there folders for each scan in that subject and then all the files for each of those scans. In this little package of fNIRS preprocessing Matlab code, I've included some demo files of the different ways you might structure your data that then we can play around with and you can practice to make sure that you're using this in a way that makes sense. For instance, you can have a study that scans single subjects, one person at a time, and then just one scan from them. So if we go into this, we have our separate subjects, and then each of those subjects is the raw data that came out of the machine. Whereas this one is a study that was done on single subjects, but with multiple scans per subject. So if we dive into that and see one subject's folder, there are now 5 different scans for that subject, and only in there are our raw files. And then finally, it's possible to preprocess hyperscanning files with this code. Hyperscanning, of course, means you had 2 people running at once. And so in this demo folder, we have dyads. The folder's laid out by dyad level, and then each scan within each of those dyads, and then within each scan, you have subject 1 and subject 2, where their raw data files live. So when you are about to run this preprocessing script, make sure that you organize your files in that sort of way and that you are labeling each subject with a consistent prefix. So for this study, this was our Minerva study, so each subject is pre-labeled min and then their subject ID, and it's going to have appended which task they were doing. This doesn't matter so much. What we care about is that some study identifier is at the beginning of these subject code or these subject names, and that it's the same for every subject. That's because this code is going to be looking through this data file and identifying anything that has this prefix and ignoring anything it doesn't. So if you're naming some of your data files some things and some of the data files something else, it's not going to be able to pick up all of the data files. They need to be consistently named. Likewise, if you're running a study with multiple scans, make sure each one of those scans is also named with the prefix. Like you don't want to say ST101 but then just rest vid1 vid2, because again this code is going to be diving into here, asking if this is a directory of multiple scans, and if so, then looking within this directory for any file that has the data prefix. This enables you to put other files into this folder if you want, things like stimuli timings or behavioral text files, whatever. So you just want to be able to delineate your neural data from that by putting this prefix at the beginning. So that means don't use this prefix for any other files you want the program to ignore. And again, for dyads, make sure the dyad number has some sort of prefix. Any individual scan within that dyad has a prefix, but then you can leave the subject names within each scan as subject 1 and subject 2, because that will always be the case if you are running a hyperscanning study with an IRX machine. Okay, so when you are running this, you essentially just type in what your data prefix is, make sure it's a string that you're putting in. So if you were typing it, you would say like that instead of just st. And then you have this Boolean argument at the end which just says, should this script consider our files to be hyperscanning or not? Put in a 0 if you ran a single subject experiment, put in a 1 if you ran hyperscanning. So the first thing this file does then is it looks for some directory for your data to live in. So you can be in any current path right now. This will just go open up a little dialog window so that you can choose your directory that your data lives in, like this. When you are choosing the directory, you will need your data structured into a single folder in which all your subjects are their own folder, and then of course within each of those is either the scans or the files. So when this pops up when you run the function, you want to single select your raw data folder and then hit open. If you double select it, you'll dive into that directory, and then that's not what we want, so make sure you hit cancel and do the single select of the raw data directory that you want. And then it will also then ask you for a probe info file. Like we discussed before, a probe info file is something that you can make previous to your data collection that essentially tells the NIRS machine and our analysis scripts what layout was your channels in and what was their spacing and things like that. If you make it prior to data collection and then put it into your probe montage, every single scan you do will automatically have this probe info file loaded in. If you don't include that in your montage during data acquisition, that's okay, but you then have to go make a probe info file and make sure it's somewhere that you can find. You will need the same probe info file to apply to every scan that you do in this study. So you can, if you've made a probe info file before acquisition and then they are automatically loaded into each subject, they'll be named differently depending on what date and time you ran this study. But that's okay for this script. You just need to select one of those, and so long as every other subject is built on the same arrangement of channels, then that probe info file will work. But the script will break if any of your subjects are accidentally having the wrong montage. And we'll go over in a second how you figure out if that's happening. So you would just select any of these probe info files. I can open this subjects, I can open this subjects, whatever, and hit open, and then you've loaded in your probe info file. From there, the next step that this script does is it extracts NIRS data. So let's go into this function that it then performs. So this is essentially just diving into your raw data files here. The most important ones it's going to be considering are this file, .wl1, .wl2, and .hdr. .wl1, if you look at it in a text editor, you can see that it's a really large file of many, many numbers. This is our raw data that's coming out of the machine for wavelength 1. Each column is a different channel, meaning source detector pair, and each row is a different time point that it recorded. So each picture it takes while it's scanning is each row. You'll notice that this is much longer column-wise than the probe layout you gave it originally. Like in this study, there's supposed to be 35 channels, and this is way more than 35 columns. That's because these files are recording not just the channels you told it to pay attention to, but every possible pairing between each source and each detector. We can know that by opening the HDR file. This has a bunch of information about like the sampling rate of your data acquisition, the date, and then what is relevant to what we're talking about right now, the SD mask. So this is saying that in that wavelength file, say column number 95 is the pairing between source 8 and detector 11. This little mask at the beginning says, okay, but which of those pairings do we care about? If we look at sources as rows and detectors as columns, or sorry, the other way around, sources as columns, detectors as rows, it wants us to pay attention to source 1, detector 1, source 2, detector 1, but not source 3, detector 1, or any of the later sources paired to detector 1. Likewise, we care about source 1, detector 2, and source 3, detector 2, but not source 2, detector 2. So when we are doing this preprocessing stuff, we need the wavelength 1 file because it has our raw data, but then it'll pare it down. Wavelength 2 looks similar, it's just all the raw data for wavelength 2 instead of wavelength 1 in our recording. So this extract fNIRS data file is just extracting all of those values we care about from the wavelength 1, 2, and HDR files. That's the header file, is what HDR stands for. Then once preprocessing fNIRS has all of the data we care about, the second step it does is identify and remove bad channels. So we talked a little bit about in data acquisition that it can occur where some channel is just going to be too noisy with no good data in it. That would be because too much hair was in front of the optode, it wasn't making good contact with the scalp, or it could be oversaturated, in which case too much signal is making it to the detector and it's freaking out. And that's what this function does. It just identifies which of those channels are going to be bad and won't contain signal that will be good for our analyses. If we look into that remove bad channels file, it's doing two things. At first, it's looking for, are there any NAN points in the time series of every channel? NAN would be happening if your time series is saturated at all. That won't happen, I don't think, ever really with our NIRS Scout machine, because it has a pretty good dynamic range. You can set the gains from anywhere between 1 and 8. And so you can turn down the gain level a lot to accommodate pretty high strength signal. But if you're using a NIRS Scout, or sorry, a NIRS Sport, that has a less wide, I guess, a thinner dynamic range, meaning there's fewer gain levels it can adjust to. So if the signal is too strong, then you're more likely to find saturated channels in a NIRS Sport acquisition. If there's just like a couple seconds or a couple snapshots of NAND values, that's okay. We can fill that in pretty reliably. Like, we can guess what the missing data points would be if there's just 2 or 3 points of NAND. So what this function is first doing is it's looking for if there's any NAND values in any of the time courses of each channel. If there are, it's going to ask, okay, is this a short little blip of NANDs? If so, we're going to interpolate, and that's what that in-paint NANDs dependency is doing. It's just interpolating what those data values are most likely to be and then inserting them into the data file. However, if the NANDs run on for a really long stretch of time, like several seconds, then we start losing the ability to fill in what those data points are, because there's room for the brain activity to be moving around in various ways in that time window that we don't know about. And so in that case, it's just going to say, all right, this is a bad channel because I can't reliably interpolate what these values should be. So it will look for those long lengths of NANDs, and if so, mark that channel as bad. Second, it then looks for where data might be present but noisy, in which case it's not, there are signal values, but there's no brain values that we care about. And to do this, it's using a process called the quartile coefficient of dispersion. Remember when we talked about what preprocessing does is you want to know about the frequency spectrum of your signal, and a good signal would have many frequencies in the sort of slow wave area and many fewer frequencies in the high frequency range of your signal. And so this is just checking, is that true? Essentially, it kind of compares the number of frequencies in the bottom quartile of that range to the number of frequencies in the third quartile of that range. A larger QCOD means cleaner signal. That means kind of the balance of the lower quartile to the higher quartile is much larger. I've set this default threshold to be 0.1. So if you care more about what this value is, what this math is like, I put just a link to the Wikipedia page in the Preprocessing fNIRS window. This also, the comment here says how many NaN points we're going to allow before we mark something as bad. So you can change that if you want as well in the remove bad channels file. It's dependent on what the sampling rate is, and I'm just essentially saying I'm going to allow 2 seconds of bad NaNs before I mark it as bad. So however many NaNs that ends up being multiplied by the sampling rate, that's what we'll look for. Anyways, our threshold default for this QCOD is 0.1. This can be changed as well in the remove bad channels. I think it's either, yeah, you can put that in as an argument here. So feel free to change these values if you want to allow longer periods of NaNs or a different QCOD threshold. You would change it to less than 0.1 if you kind of want to allow there to be greater noise in the signal, or you would change it to larger than 0.1 if you want a more stringent threshold. And while it's doing all that, it makes a channel mask that's just a series of zeros and ones for each channel. In your scan, ones will be, this is a good channel, 0 is, this is a bad channel. Once it spits out that channel mask, boop, we then convert to the .NIRS format. This will be less relevant to you if you don't care about using Homer 2 for any other things. This is just their data structure that they use in their set of scripts. And on top of the NIRS variables we already extracted, there are some other things that it pulls out, mainly things from the probe info file. So this is just preparing those if you want to be able to use them. And then finally, we do our preprocessing, filtering, and hemodynamic conversion. All of this is just getting our raw data ready to go into this folder. This is the function that actually does the preprocessing. So let's look into that. Here. So this would be our filtering pipeline. If you ever want to change what preprocessing steps you do, this is where you would do it in our code. In our pipeline right now, we have it set up to do motion filtering. Right now it's detecting where motion is happening. It's then correcting that motion using a PCA algorithm. It's converting then to concentrations, and in that conversion it's also doing a bandpass filter. So that's what these frequencies are. That's the low and high bandpass cutoffs. And then lastly it z-scores and saves our data in both chromophores, total chromophores, and then the Z versions of all of those. And so you can feel free to change these parameters if, say, you want a larger window around the HMR or around the motion artifact that we're detecting, if you want a different bandpass filter. I would recommend diving into these functions in the Homer 2 script to learn what they're doing. They're pretty simple. Okay, and so that is all the sub-functions of this. The last thing that happens then is just to save these outputs. We're saving both the 6 versions of the chromophores as just kind of a Matlab structure that you can then open and look at all of these as matrices in Matlab, and then as the NIRS format, which saves the raw or the preprocessed data as well as these other information bits about like what your probe structure was and stuff like that. So now let's run it. Make sure that preprocessing fNIRS and the support functions are somewhere on your path. Either you are sitting in the folder that has it, or this is saved to your default path in Matlab. Like this instruction says, make sure you include these 2 inputs in order to run stuff. Let's try out the single subject, single scan test group first. So we would say preprocessing fNIRS. In this one, our prefix is min, so I would say min. And then this is single subject, not dyads, so 0. Then hit enter. So it's asking for our folder of data. Make sure you single click this and hit open. Now it's asking for a probe info file. Go into some subject, find their probe info file, and click open. It'll then give you a readout of how many subjects it's done so far. And at the very end, it'll tell you how much time happened. So that was almost 2 seconds per subject, and this was a study where the scan lasted about 15 minutes long, but there were only 1 scan per subject, and then there was only about 20 channels per person. This will take longer if there's many more channels on someone's head, it's lasting longer, or if you have more scans. So this function is set up to save your preprocessed files as a preprocessed files directory within your raw data directory. It's at the same level as each one of your individual subject files. If you dive into that, you have a folder for each of your subjects, and then the 2 output files that we made for the raw data, or the preprocessed data. Say if we were to open this one, it would give us our oxy, deoxy, total oxy, and then the z-scored versions. And if you look at these matrices, you have channels along the top, 20 channels in this data set, and then time points in rows. And there was 6,711 data points recorded at about 8 hertz, I think this one was, so whatever that number is, number of seconds. You'll notice in this output file that there are some columns marked with nans. So these were our bad channels. The entire time course will be marked nan if this channel was bad, so make sure when you are analyzing your data, you're prepared to handle nan values. Any channel that has numbers in it is a good channel. Now say you were running this function on a big data set, and then something happened halfway through the preprocessing. Maybe one of your subjects had the wrong montage, and so then the function errors out. You can fix whatever caused that and then run it again, and it won't do the entire preprocessing batch again. It's set up to identify if this folder already exists, and if not, it'll pick up where, or if it doesn't exist, it'll do everything. If it does exist, it'll pick up wherever you left off. So say if we were doing Minerva 201 through 220 and something broke at 211, we would have files for 201 through 210. Fix whatever's wrong with 211, and then it would only preprocess 211 through 220 and add it into this preprocess files. So that'll save you some time if you ever need to debug something. Speaking of debugging, there are a couple notes I put in here that I've run into that are kind of common errors and reasons for this to not work. One thing is you might get some warnings pop up. I've gone ahead and forcefully suppressed these in, where is it, the fNIRS pipeline here, warning off. You can turn that back on if you want, and then these warnings just mean in our filtered data, sometimes the emotion correction algorithm will force raw data points to being negative. Normally, negative data points are a bad thing. It means there's like noise going on. But if the motion correction algorithm is forcing that to be the case, it's okay. This intensity to concentration conversion script will give a warning if there are any zeros or negatives in the raw data in case you weren't doing this. But since we are, I'm just turning that off. If you have a particular reason to have those warnings pop up, you can turn warnings back on or delete this. I've set warnings back on at the end of this just in case you want other kinds of warnings popping up while you're running the other scripts. So just be aware of that, but if you leave that warning alone, this doesn't apply to you. And then some debugging tips. So this works right now on the demo files that I've included, and it also works on any data folder that I've personally run. I have not tested this to the same extent on a PC, and it's still possible there are some edge cases that I've missed. So things that might occur is if you get an index exceeds matrix dimensions error in the HMR motion artifact for a subject that's not the first file, make sure that the SD mask structure in the header file of that subject matches the channel structure of the selected probe info file. That means, say you ran a montage that was expecting 32 sources by 32 channels, but then you gave it a probe info file that said this data structured 32 sources by 24 detectors. That will make this poop out an error. So just be sure that your SD mask in the header file is correct in what you're expecting, and that it matches the correct probe info file that you feed into the data at the very beginning. Make sure also that any false start files, scans where it runs for a couple seconds and then might quit because you didn't mean to start recording yet or something happened like you double clicked, just remove those because that will also cause it to error out. Okay, so let's practice now for a Multiscan project. Again, this would be where you have different subjects, and within each of those subjects, you scanned them multiple times. Again, the way you input that is no different. Just make sure you're using the correct prefix for your study. Since it's still a single subject, use a 0. Now, we're not using this one, we are using this data set. So open that, go into the correct data set, find someone's probe info file in one of their scans, and open. And then you'll notice this is going to be running slower than the other data set did. That's because in this study we had 108 channels instead of 20, and then there are 5 scans instead of 1 scan per person. So this will run slower, but I've still never had the problem where this would run or take longer than some MRI pre-processing pipeline. When I did this study in a batch of 60 subjects, it took me about 20-30 minutes. And so when this finishes running, we can see exactly how long it took. It'll probably be about a minute in this run. Let's see. Almost there. There it is. Yep. 58 seconds. Cool. And now again, we have our preprocessing files folder in our larger raw data folder. We have a folder for each subject, for each scan, and then the preprocessed data. Finally, let's do a demo for dyads. In this study, we had some dyads. There were 2 scans per dyad, and then with each of those scans, we have our subject 1 and subject 2. So this is where we actually do have to put something different into the command for preprocessing fNIRS. We have to include the prefix, the correct one for this study, and then use a 1 instead of 0. Let's close this, make sure we're picking the right data folder, the right probe info file, and then it preprocesses it. For this study, there were 35 channels on each person's head, 2 scans, and the scan length was variable. So some of these dyads will preprocess much faster than other ones if one dyad took much longer in their study. Cool. So it took about 21.5 seconds. There's also in this preprocessing script something else we haven't talked about yet, and that's this test QCOD file. This is not necessary for preprocessing data. The only reason you might use this is if you thought you might want to change what the QCOD threshold is for removing bad channels, but you're not sure what that threshold is. So this helps you visualize the frequency spectrum of each of your channels in someone's data to see, okay, I think this is what a bad channel would look like. Is it being marked as bad? So let's go into what this does. It takes 2 arguments, the QCOD threshold you're testing, and then whether you're going to suppress the plot or not. This QCOD threshold, again, would be a default of 0.1 in the remove bad channels file. If you increase that, you would be making it more stringent. And then suppress plots. What this function does is it spits out a channel mask. That's that series of zeros and ones saying if this channel should be marked as bad or not. 0 is bad, 1 is good. And then it also opens up a little plot of the frequency spectrum for every channel. You can look at each one of those plots, though if you've done this a few times you know what the plot looks like, or if there's tons and tons of channels you might want to suppress that because it can take a while to open up all those plots. So in that case you would just put 1 to suppress plots or 0 to let plots happen. So let's test this out. We'll test our default first to see if we like that, and then we're not going to suppress so we know what those plots look like. First, it opens up a little dialog box to say which subject should we be testing this on. You need to pick a data folder that contains the NIRS raw files. So for instance, if we were in our dyads folder, pick some subject level data folder. If we're in our single subject scan with 1 scan in this per subject, here's where our raw data is, so pick this folder. Or in our multiscan single subject, go into 1 subject's data, 1 scan's data, and pick this folder. So let's test it on, say, this subject in our dyads study. So it spits out our channel mask. It marks a few channels I think should be bad, and then our figures, that's the frequency plot for each channel. So say it thinks channel 33 is bad, since this is columns 31 through 35, 31, 32, 33, 34, 35. We can look at the spectrum for each of these channels. This is 35. It's kind of messy, but we still have kind of a nice, strong dominance of low frequency stuff, so we could say this passes. If you thought this is too messy, you could lower your threshold a bit. But 34 looks pretty nice. This little bump here would just be the heart rate, which is fine because it's much slower than our cerebral data that we care about, and it's going to be filtered out anyways. But 33 is messy. This is still a larger peak than anywhere else, but these are big enough compared to this that it's our 0.1 threshold saying, nah, this is going to be too messy. We're not going to be able to recover enough good data from this. Some other channels that we can look into might be, let's see, this one is pretty bad. There's not even any peak whatsoever at the lower frequencies. This is pure noise. So you would definitely be wanting to reject this one. 16, 17, 18, 19, 20, 21, 22, 23, 24. Yep, it's being rejected. So there are some points where this is being rejected too because the heartbeat is so strong compared to this one. You can decide if you want that to be the case or not. Let's see. Another one would be 1, 2, 3, 4, 5. Now 6, say we think this is too messy, but it's accepting 6, right? 1, 2, 3, 4, 5, 6. So we can change our threshold a little bit. Let's try that out and what happens. Sorry, I think I misspoke before where we want to raise the threshold to make it more stringent. Let's try that out. Yep. Okay, so now it's getting a little bit more stringent. We have more channels being removed. This time we're saying, yeah, 35 we want to take out. And 33, 1, 2, 3, 4. And this would be 16. So now we're taking out this 35 channel. So again, push the threshold higher if you want it to be more stringent on what it counts as a good channel or not. And now I'm going to suppress the plot so I don't have to keep closing all of that for you. But let's say, what happens if we do 0.5? And that's like half our data is being removed now. Is bad. It's probably removing anything that had any sort of cardiac signal in it whatsoever. So that's how you use the testing QCOD function to kind of narrow in on which QCOD threshold you want to use. And that is the broader demonstration of how to use this preprocessing stuff. If you find any bugs in this, please let me know in some capacity, either like do a little update on Git or just like email me or something and I'll make changes to it. And then if you're using the Python version, go watch the Python video.